Good morning all of you. In the last class we have completed our chemical characteristic of water, and today we are going to discuss about the biological characteristic of water. As you know, bacteria are the minute single cell organism having no green material, that is chlorophyll, which will help them in manufacturing their own food. They are present generally in raw and contaminated water. They are generally of two types. One is non-pathogenic bacteria, second one is pathogenic bacteria. So, non-pathogenic bacteria means which are harmless to the human being, animal and crops. Such bacteria are called, these are sometimes under certain conditions these are also beneficial. So, those type of bacteria are called non-pathogenic bacteria. On the other case, some bacteria are harmful to the human being, causing waterborne diseases such as cholera, typhoid, etc. and those are called pathogenic bacteria. Most of the bacteria present in water require oxygen for their survival. So, these bacteria, they will consume dissolved oxygen from the water and decompose the organic matter present in water. So, depending upon the oxygen necessities of the bacteria, they may be classified as follows. The first one is aerobic bacteria. So, these bacteria require oxygen for their survival. Second one is anaerobic bacteria. So, those bacteria can survive in the absence of oxygen. Third one is facultative bacteria. So, which can survive with or without oxygen. The combined group of pathogenic and non-pathogenic bacteria is designated by Bacillus coli or B. coli group. Okay. And this group of bacteria are present in intestine of all living warm blooded animals. And also it should be noted that practically all natural waters contain some variety of bacteria. Even rain before touches the surface of the earth collect dust and bacteria according to the presence of industries, climatic conditions, etc. Therefore, these bacteria can be tested by the following test. One is total count or agar plate count test. Second one is B. coli test. So, let us first discuss about the total count or agar plate count test. So, in this method, total number of bacteria present in milliliter of water is counted. So, first we have to take 1 ml of sample of water. Then this will be diluted by adding 99 ml of sterilized water. Then from the diluted water we will take 1 ml of diluted water and it will be mixed with 10 ml of agar of gelatin, which is a culture medium. Then that mixture is to be kept in an incubator for 24 hours at 37 degrees Celsius or for 48 hours at 20 degrees Celsius. After this period, the sample will be taken out from the incubator and colonies of bacteria that are formed will be counted by means of microscope. Then the product of the number of colonies and the dilution factor will give us the total number of bacteria per milliliter of undiluted water sample. So, this is all about the total count or agar plate count test. Then next we will move to bacteria coli or B. coli test. This test is also called as E. coli test. It has two parts. First one is presumptive test, second one is confirmative test. So, in the presumptive test, definite amount of diluted sample is to be taken in standard fermentation tube which contain lactose broth as culture medium. So, first the sample of water is to be taken in standard fermentation tube containing a culture medium. Then the fermentation tube is to be kept in an incubator for 24 to 48 hours at 37 degree Celsius. Then it is observed that if some gas is produced in the fermentation tube, this will indicate the presence of B. coli or vice versa. After this confirmation test, after this presumptive test, for confirmation test, some sample of presumptive test is to be taken and placed in another fermentation tube containing brilliant green lactose bile as culture medium. Okay. First we will go for the presumptive test, and if we will observe that there is some presence of B. coli, then we will go for the confirmation test. And in this confirmation test, we have to take the sample from the presumptive test, which will and it will be kept in another fermentation tube containing brilliant green lactose bile as culture medium. Then this will again kept in an incubator for 48 hours at 37 degree Celsius. And if there is formation of gas in the tube, it will confirm the presence of B. coli and the water is now unsafe for use. Okay. So, this is about your B. coli test. Next technique is membrane filter technique. So, this is very simple method, and in this method the sample of water is to be filtered through the sterilized membrane of special design which will retain the bacteria on the membrane. Then this membrane is to be put in contact with culture medium, that is M. indose medium, and in that it will be M. indose medium. Then it will be kept in the incubator for 24 hours at 37 degree Celsius. Then the membrane after incubating is taken out and the colonies of bacteria are counted by means of microscope. So, this is about the membrane filter technique, and with this we have completed our bacteriological characteristic, and in the next class we will go for the sources of water supply. Thank you.